this video is going to be about the structure of proteins. So proteins are made up of the elements carbon, hydrogen, oxygen, and nitrogen. So this is the new element that we did not see with carbohydrates or lipids. And some contain sulfur and phosphorus. So first I'm going to talk about the functions of proteins, and there are many of them. First of all, proteins are the structure of our cells, bones, cartilage, muscle tissue, collagen, and our hair and nails. So they're a lot of structure. Our hormones are proteins, and hormones are chemical messengers. You're probably familiar with testosterone, estrogen, maybe progesterone you've heard of. Antibodies, which are the molecules that are part of our immune system that fight disease, proteins too. And enzymes. We're going to have a whole video lecture on enzymes specifically, but enzymes allow complex chemical reactions to occur at a faster pace than they would normally, without the presence of enzymes. And so pretty much every biological reaction that happens in our body, it requires its own specific enzyme to catalyze that reaction. So now let's talk about the structure. So now with this unit, video, we're back to monomers and polymers. So proteins are polymers of this building block, this monomer called an amino acid. It would be really nice if they called it a mono something, but unfortunately scientists didn't name it that. But it's referred to as an amino acid. The carboxyl group, which you saw in the fats video, is a carbon double bonded to an oxygen with a hydroxyl group. Now, sometimes it's drawn just OH, but there would be a bond, and there is a bond between the oxygen and hydrogen here. It's just sometimes that chemists don't draw that. They just assume that we all know that. There's a central carbon atom bonded to a hydrogen, and then there is what's called the variable group. There are 20 different things that go here. Why R was chosen to represent the variable group, I'm not sure. At some point somebody probably told me, but I don't remember. And then we have the amino group, the nitrogen bonded with the two hydrogens. So lots of molecules that we've been studying have a carboxyl group. What is unique to the amino acids, and therefore the proteins, is this nitrogen bonded to two hydrogens, this NH2. So that's what you're going to look for. So an amino acid is the monomer or building block of proteins. This is our amine group. That's the carboxyl group. Okay, like we just said, make sure you know those for tests and quizzes. Now, how do we get long polymers from that monomer? Well, we bond them together by dehydration synthesis. So, dehydration synthesis happens between the amine group of one amino acid and the carboxyl group of the other amino acid. And obviously, if we're going to make water, the OH comes off the carboxyl group, and one hydrogen comes off the amino group to make a water. And when you have two amino acids bonded together, you have what is called a dipeptide, and many together is called a polypeptide. Now, this diagram is going to show you two amino acids, and they're showing you the OH from the carboxyl group and the H from the amine group coming together to form water, and then a bond forming between this carbon and this nitrogen. Now, the molecule is called a dipeptide. The bond between what were once two monomers right here is referred to as a peptide bond. And you're going to have to be able to find the peptide bond in diagrams, or you're actually going to build one of these molecules. And it's always between the carbon that is double bonded to the oxygen and the nitrogen that is left over from the amino group of the other amino acid. So here we go. We have two amino acids. We have water forming. And we have the carbon and nitrogen bonding to each other. This is the peptide bond, and what you just saw, of course, was our old friend dehydration synthesis. So now I want to talk about the three levels of structure of protein. So the first level of structure is referred to as the primary structure, and that is the amino acid sequence. Now, the primary structure can vary based on the sequence of the 20 different amino acids that exist in nature and the length of the polypeptide chain. The smallest protein that's a human protein, I believe, is insulin, and if I recall, it is 52 amino acids long. That's actually relatively short. Now, if you imagine that there are 20 different amino acids and all kinds of different lengths and sequences of amino acids, then you'll understand why there's a wide variety of proteins that can possibly be made and are made in nature. Now, be careful because students sometimes want to say, when they're comparing one protein to another, that it's made of different types of amino acids. And that's just not true. All of the proteins in nature are made of the same 20 amino acids. Now, some proteins may not have all 20 in them, just like you may have words in the English language that don't have all of the letters of the alphabet, but all of the words in English are made with the same alphabet. So think of the 20 amino acids like the protein alphabet. Now, once you have the primary structure built, we have what is called the secondary structure. So those chains twist in space and often form two common shapes. One is the alpha helix shape, which you can see here, and it's the same shape of a DNA molecule, although a DNA molecule is a double helix. And sometimes you get what is referred to as a beta pleated sheet, and it's kind of folded back and forth. It reminds me a lot of those fans that we used to make in elementary school with a piece of notebook paper on a hot day when the air conditioning wasn't working, and you would fold the paper back and forth and back and forth. That's very similar to the shape of a beta pleated sheet. Now, once you have the secondary structure, either the beta pleated sheet or alpha helix will fold further based on which parts of the chain are attracted to water and which parts are repelled by water. The hydrophobic portions fold away from the water inwards, and then the shape is stabilized by crosslinks between the variable groups of the amino acids. So remember that R group. Now, every time you have the same primary structure, you will get the same secondary and tertiary structure. And if you make a mistake when you're building a protein, well, you wouldn't, but if a cell makes a mistake, and the amino acid sequence is different, the secondary and tertiary structure will also be affected. Kind of like when you're doing a math problem and you make a mistake in the beginning and then everything after that is incorrect. Now, some proteins, but not all, reach the quaternary stage of the structure. All proteins, or it's not really considered a protein until it reaches its tertiary structure. Some proteins are made up of more than one polypeptide working together. One example is hemoglobin, which is the protein in your body that carries oxygen in your red blood cells. And there are many more, but that's one that you're probably familiar with or you've at least heard of before. So the amino acid sequence, the primary structure, determines the protein's shape because it determines the secondary, tertiary, and sometimes quaternary structure. Remember, not all proteins have a quaternary structure. And a protein's shape determines its function in an organism. If you change the shape, the protein will not function anymore and do its job. So it is the difference in the amino acid sequence and the number of amino acids in the polypeptide chain that make the variety of proteins possible.